Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. Today in this video lecture, we are going to talk about blue-white screening. In the previous video lecture, if you guys remember, we talked about differential screening. Okay. Now, what is blue-white screening? What are the importance of blue-white screening and how we can perform blue-white screening? We'll be talking this video lecture. So let me clean the whiteboard and let me start. Okay, so let me write blue. Now, what is blue-white screening? It is again a screening technique in which we can detect our recombinant vector, our recombinant cell inside a media or in a neutron agar plate. Okay. Very fastly or rapidly. Now, how we can perform it? Now, what are the importance of this blue-white screening technique? Suppose you have your bacterial cell, and this bacterial cell can carry your recombinant DNA vector. And when you grow this bacterial cell in the presence of media which contain antibiotic substance, when you grow this cell, so these cells will be grow. But the other cell which don't carry your recombinant DNA vector plasmid, this bacteria will be die. But let's suppose what actually happens. Sometime, let's suppose this is the media plate. Okay, and here you can grow your recombinant DNA vector, those bacteria which can carry recombinant DNA vector. So let's suppose this is the bacteria that can carry your recombinant DNA vector. Okay, but this media is what you can add the antibiotic in this media. That's why other bacteria which don't have the recombinant DNA vector will be die. But sometime what actually happens, sometime this media have also all those bacterial cell. Let's suppose they have all those bacterial cell which can grow. Now these colony, some of them are those colony which can carry the recombinant DNA vector, and some of those colony which don't carry the recombinant DNA vector. Now, how is it possible that other bacterial cell can also survive? Because in other bacterial cell there is also a chance that in such bacteria there is also a plasmid, and there may be that some plasmid have also the resistant genes which have the ability to degrade the antibiotic substance which is present in this media. So then such technique will be not used in our daily life. Then we not use this one technique. This is actually a selectable marker techniques, or this is actually media or we can say replica plating technique, but we usually not use, and we are usually not preferable this screening technique, because the same issue are created in this technique as I told you. Because in this technique, your vector is also your bacteria having which can carry your recombinant DNA vector, and all other bacteria can also grow which have the plasmid, but the plasmid one bacteria can also grow and your bacteria can also grow. But how you will differentiate that which one colony have the bacterial cell, means your recombinant bacterial cell, which one colony? It is very difficult for us. So for that purpose, we can use the blue-white screening. That's why it is very important and it is very useful in this case. Now, how can we perform this blue-white screening? In this case, there is vector always available in the market. In such vector, we have let's suppose this is the vector. Okay. Now, in this vector, we have a reporter genes which is always present. And this reporter gene is called as beta galactosidase. Now, this is the reporter genes. Okay. And this plasmid or vector also contain antibiotic resistant genes as well, which we call that ampicillin. Okay. Now, this vector we always use this vector in blue-white screening. And somewhere at that specific site, there is the restriction site is also. Let's say there. Near the beta galactosidase or near the reporter genes, there is also the restriction site or multiple cloning site is also present there. Now when we cleave the vector with restriction enzyme, and when we apply or when we add our target DNA, so let's suppose this is our target DNA. So when we join our target DNA with this vector by cleaving both vector as well as the target DNA with the same restriction enzyme, and when we join it, so then we get the structure like that, as you can see. Now this is the target DNA. This green one can represent the target DNA having gene of interest. This one, the black one, let me remove. This black one can represents the reporter genes, reporter genes, or we can say beta galactosidase. And somewhere at that specific site we have also the antibiotic resistance genes as well, which we called ampicillin. So then we get a structure like that, and after that this is called recombinant DNA vector. Okay, recombinant DNA vector. Then we will insert this recombinant DNA vector by making the cell competent. We will insert it into the host cell. Okay, so let's suppose this is our host cell. This host cell could be a bacterial cell, or it could be a eukaryote or prokaryote. If it is prokaryote, so it will be or it must be E. coli. If it is eukaryote, it must be yeast cell. Now once we loaded the bacteria, the recombinant DNA vector inside the host cell by making the host cell competent, after that they will uptake the vector. Now then we grow this bacterial cell, or we grow this host cell in a media. So let's suppose this is a media. So we grow this cell in this media having a substrate which we called it that agar and antibiotic substance as well. So they have two things, antibiotic substance as well as agar. Agar is actually a substrate which we loaded here. Now all the bacterial cell which can carry the recombinant DNA vector will grow here, and they will show white colony. So let's suppose these are the bacterial cell, or these are the colony. Now these are the white colony, as you can see, and these white colony can show that your recombinant cells are present in each colonies. Okay, now when we get a blue colony, let's suppose these are the blue colonies. When we get the blue colonies, so then this blue colony shows that these are those means inside each colony there are those bacteria which don't carry your recombinant DNA vector. Now how? Because remember, whenever we loaded or whenever we combine this target DNA near to the reporter genes, near to this beta galactosidase gene, then this enzyme will be disturbed, or this enzyme will be disrupted, or this enzyme will be deactivated or inactivated. Now remember, when the enzyme is inactivated, then there is no beta galactosidase production. If there is no beta galactosidase production in the plate, so enzyme, there is no enzyme which can bind with the substrate. If the enzyme is not, this X-gal is actually a substrate. When the enzyme, which is the beta galactosidase, which is not produced, if it is not produced and if the enzyme is not bind with the X-gal, so there will be no blue color colony. If there is no blue color colony, it means that this enzyme is deactivated. And how it is deactivated? Because here you can put something, and this something is actually your target DNA. And white colonies appear, and we can say that this is actually the recombinant cell. Recombinant cell. Okay? Sometime what actually happen? If let's suppose you didn't put any kind of vector here, let's suppose, if you didn't put any kind of target DNA here, let's suppose in this case again you have the vector. Let's suppose this is again the vector. Okay, now again this black one can represent that these are the reporter genes, or these are the beta galactosidase producing genes, and these blue one can represent the antibiotic resistance genes. Okay, but there is no, there is no target DNA can bind here. Then when, remember, when we grow this, when we insert this, this is not a recombinant DNA vector, this is just a vector. When we insert this vector into the host cell, and when we grow this host cell into the media, then what actually happen? We get the blue colony. Why? Because the gene is not disrupted and this gene is activated. Now when the gene is activated, then what actually happen? This beta galactosidase is always present in lac operon, means it is always present in lac operon area, and lac operon can actually produce the beta galactosidase. Usually lac Z genes can produce beta galactosidase. Okay, then what actually happen? If we grow the bacterial cell into the media, then the bacterial cell can show, or the colony will be appear in a blue color. It means that beta galactosidase is produces, and when the beta galactosidase is produces, it means it is not disrupted. Why it is not disrupted and why it is activated? Because you cannot put your target DNA. When you not put your target DNA, that how it will be deactivated. Now let's suppose it is activated in form. When it is activated, so it will be reacted, means this beta galactosidase will be reacted with X-gal. X-gal is the substrate, so when substrate and enzyme are reacted, so it can produce a color in the form of colony. So we get a blue color in the form of colony, as you can see. And it can show that these are actually the cell which don't carry the recombinant DNA vector. Okay, so this is all about the blue-white screening, and I hope you guys get idea about the blue-white screening. Inshallah, in the next video lecture we are going to talk about expression screening. Okay, so please stay tuned and continue to watch the video. Thank you so much.